Hello, everyone. So today's lecture is going to be on metabolism, on energy and enzymes, Now, I would suggest that you copy down this illustration in your notebook. This is something that you will have to know about. So go ahead and copy this somewhere in your notebook. All right, the flow of energy through life. So life is built on chemical reactions. As we see, ultimately, all of our energy comes from the sun. The sun inadvertently gives energy to the plants, right? Or herbivores. They take in the sunlight and in return they make glucose for our herbivores. Then we have our carnivores, such as the lion. And the lion then obviously will eat the giraffe and it gets its energy or it retains its energy. So it's built on all of these chemical reactions, a circle of life. Now, the chemical reactions of life is known as metabolism. Metabolism means forming bonds between molecules and breaking of the bonds between molecules. It's the way our body functions, or it's all of the chemical reactions in our cells. Metabolism. Whenever we form bonds and whenever we're making something, that's known as dehydration synthesis. Dehydration synthesis, which is also termed anabolic reaction. So another term for dehydration synthesis is anabolic reactions. Whenever we're breaking bonds between the molecules, that function is known as hydrolysis. And that's a form of a catabolic reaction. So I'm breaking something apart, catabolic reactions. So make sure that you guys know these terms. So we're going to go with catabolic and anabolic. Anabolic is making things, catabolic is taking them apart. So here's our example. As we recall, whenever we're forming a molecule through the process of dehydration synthesis, so for example, if this is glucose or this is our sugar, sucrose, right? In the function of making a double sugar, I get one molecule, one monomer. I place those together to make this double sugar and water gets released. Now, on the opposite hand is hydrolysis, also known as digestion. And this is when I have a large molecule, I add in water, and in return, I break them apart. So dehydration synthesis and hydrolysis. So putting things together and taking them apart. So remember, if I put them together, water gets released. If I'm breaking it apart, I need to add water to restore the bonds. Now, some chemical reactions release energy. These are known as exergonic reactions. So make sure that you guys know that. Anyone that is releasing energy is called an exergonic reaction. An exergonic reaction occurs when we are digesting polymers due to the process of hydrolysis or catabolism. So anytime I'm digesting, energy gets released. Why? Because I'm breaking bonds apart. Whereas some chemical reactions require an input of energy. Some of them require an input of energy. Those are known as our endergonic reactions. So this is when I'm building my polymers, when I'm building something. Okay, I built that through the process of synthesis or dehydration synthesis as another form of anabolism. So once again, whenever I'm trying to retain energy, that's a form of endergonic reactions because I'm actually building something together. Whenever I'm breaking apart the molecules, that's known as catabolism or hydrolysis, and that's an exergonic reaction because energy gets released. All right, so in this slide here, we're looking at an endergonic versus an exergonic reaction. So to read these graphs, as we can see, let's say here's our molecule that's going to be broken down. So in an exergonic reaction, energy gets released. As we can see here, this is the amount of energy needed to break apart the bonds. So this molecule is broken down into these three smaller molecules, and this here means that energy is released, or that's our change of G, which is our change of free energy, or the ability to do work. Whereas in an endergonic reaction, here's my reactant, and in the end, I'm building these molecules. So when I build these molecules, I retain the energy. So exergonic is when the energy gets released, Endergonic is when I am maintaining or I'm building the energy. So here's a good video on what triggers a chemical reaction. Go ahead and watch this. This video is going to talk about the difference between the endergonic and the exergonic reactions. Endergonic and exergonic reactions. All right, so same thing, like we said, our energy and life. Organisms require energy to live, just like me and just like you. So where does that energy come from? Well, the energy comes from a coupling of our reactions. So these two reactions basically need each other, our endergonic and our exergonic reactions. So what's going to happen is I'm going to eat food, right? And then in the process of digestion, I'm going to break apart the food that I eat, and in return, I get energy. Once I break apart the molecules to their monomers, these monomers here then get reused in the process of dehydration synthesis, and then energy gets restored, and I build molecules. The energy that we are used to, that our body needs, is called ATP. ATP. And ATP is the energy for your cells. ATP stands for adenosine triphosphate. Adenosine triphosphate. So ATP. And it's a high energy compound which is used to drive the metabolic reactions. It's constantly being generated from adenosine diphosphate, or ADP. ADP. It's composed of an adenine group, a ribose group, and 3 phosphate groups. So the adenine group is actually a nitrogen base. So it's made up of a nitrogen base, ribose, which is a 5-carbon sugar, and then our 3 phosphate groups. Okay. It's a coupled reaction. So energy is released by the exergonic reaction captured in ATP, and then ATP is then used to drive our endergonic reaction. So let's go ahead and see what that's about. So this is what our ATP molecule looks like. So ATP is made up of our adenine group, our ribose sugar, and then our 3 phosphate groups here. Now, as we see with this ATP cycle, like we said, it's endergonic and exergonic reactions. So they're coupled, meaning that they depend on each other. So exergonic and endergonic reactions. So what does that mean? If I look at ATP, right, the creation of ATP from ADP requires the input and energy from other sources. So it has a positive delta G, or it has energy, right? So when I do work, the energy located here on the phosphate group is unstable, and the energy gets released. Okay, so ATP will now form ADP. So energy gets released in the form of the exergonic reaction. Now I gotta build ATP again. So the hydrolysis of ATP released previously stored energy, which allows the change in free energy to do work, and it has a negative delta G. So now we add our phosphate group to restore the bond, and then we go back to ATP. So you go ATP, I do work, I lose a phosphate, I become ADP. I add the phosphate back and I become ATP. So it's exergonic, endergonic, exergonic, endergonic. So I'm releasing energy and I'm making energy, releasing and making. My system does that all day long to keep me alive. Here's another example of a coupled reaction. So let's take a look at lifting weights. So as we can see here, myosin, which is the fibers found inside of our muscles, assumes its resting shape when it combines with ATP. ATP is going to split the ADP and the phosphate, which causes the myosin to change its shape and allows it to attach to the actin molecule. Now, the release of ADP and phosphate causes myosin to again change its shape and pull again, stacked in, generating the force of motion, which then causes your muscle to contract. So energy gets released, energy gets absorbed. All right, spontaneous reactions. So if the reactions are downhill, why don't they just happen spontaneously? Well, the reason why is because the covalent bonds are stable. So why don't polymers such as carbohydrates, proteins, and fats just spontaneously digest into monomers? Well, they need something to actually break them apart into something that's smaller, something that's smaller. In order for us to get energy, we have to give up some energy. And this is known as activation energy. Activation energy. So please make sure that you guys highlight this term, activation energy. This is a term that you're going to have to know. So activation energy means breaking down large molecules requires an initial input of some energy. So large biomolecules are stable. They must absorb energy to break the bonds. So let's look here. If I look at wood, right? If I needed to make a fire, obviously I'm going to release all of this energy. To release the energy from the wood, I have to actually input a little bit of energy. So I have to start the fire in order to get a larger fire. So I light my match. I then light my wood on fire, and then the energy stored inside of the wood gets released as heat, as we see here. So the amount of energy needed to destabilize the bond of a molecule moves the reaction over the energy hill, as we see here. This here is called our activation energy. Activation energy meaning that this is the amount of energy needed to break the bond apart. This is the amount of energy that's needed to break the bond apart. Now, we want to be able to reduce that energy. So like we said, our bodies are very, very smart. They're so smart that they always want to obviously help us work better, work less. So work not as hard, work smarter, not harder. We have what are known as catalysts. And what a catalyst does is it reduces the amount of energy to start the chemical reaction. So if I look at this reaction here, so let's say I ate sucrose, and I know that this is the amount of energy that's needed in an uncatalyzed reaction. It's a lot of energy to break down this sucrose molecule into two different molecules or to its smaller molecules, glucose and fructose. Now, when I add a catalyst, notice that now I don't need to break as much energy to break the molecule apart. So it takes a lot less energy. So what's a cell to do to reduce that activation energy? We get some help from these molecules, which are called enzymes. And what enzymes do is they drop the activation energy, or they lower the amount of energy needed to break the molecule apart. So what are enzymes? Please know this slide. Enzymes are biological catalysts. They are proteins. So if they are proteins, proteins break down to which macromolecule or to which monomer? Proteins break down to, I'm hoping you said amino acids. So proteins break down to amino acids. Now what they do is our enzymes help to facilitate chemical reactions. They increase the rate of the reaction without being consumed. So meaning that when your enzyme breaks something apart, it doesn't fall apart. It reduces the activation energy, and it doesn't change the free energy that's released or required. They're required for most biological reactions, and enzymes are highly, highly specific. We have thousands of different enzymes in cells, and they help to control the chemical reactions in our body. We make thousands of different types of enzymes. Some of them are super important, and some of them not so much. Some other terminology you need to know are substrates and products. So a substrate is the reactant what binds to the enzyme. So the substrate is the molecule that's going to be broken down. It forms what's known as an enzyme substrate complex. So the substrate's going to attach to the enzyme, and in return, we're going to make what is known as a product or the end result. So let's look here. Here's my enzyme. This substrate sucrose consists of glucose and fructose is bonded together. The substrate binds to the enzyme, forming an enzyme substrate complex. Water gets added. I add water once again to restore the bonds. Then the products are released and the enzyme is free to bind with another substrate. Let's go back to number 3. The binding of the substrate and the enzyme places stress on the glucose-fructose bond, and then the bond breaks, which then releases these molecules. Okay, so water gets added, enzyme gets broken up. The enzyme breaks the substrate apart, and once it's done, it's able to go ahead and look for another sucrose molecule. And it'll continue to do that until there's no more. So the enzyme plus the substrate is going to give us a product. Sucrase is the enzyme that breaks down sucrose. It binds to sucrose and breaks the disaccharide into fructose and glucose. DNA polymerase is an enzyme that builds DNA and it adds nucleotides to a growing DNA strand. So we have enzymes that break things apart and we have enzymes that put things together. Enzymes and substrate form what is known as a lock and key model. It's a simplistic model of the enzyme reaction. It's a 3D structure of enzymes that fix the substrate. The active site is the enzyme's catalytic center. It's a pocket or a groove on the surface of globular proteins, and the substrate fits into the active site, just like a lock and key. This area, as we see right here, is the active site. Active site. And the active site would be located here. Active sites are highly specific to the substrate that it's going to break apart. It's also known as the induced fit model, so it's also synonymous with the induced fit model. It's a more accurate model of the enzyme action. This 3D structure of enzyme fits the substrate. As the substrate binds, the enzyme changes shape, leading to a tighter fit. It's called a conformational change, which then brings the chemical groups in position to catalyze the reaction. So as we see here, here's our enzyme. These little circles are going to be our amino acids. Why? Because enzymes are proteins and proteins break down to amino acids. We have our active site here, and as we said, the enzyme will fit perfectly, and then the active site will go through this conformational change, which will then break apart the substrate. So how does it work? There's a variety of mechanisms to lower activation energy and speed up reactions. The active site orients the substrates in correct position for the reaction. The enzyme brings substrate closer together. The active site then binds the substrate and puts stress on the bonds that must be broken, making it easier to separate the molecules. Just like we see here. So here's our enzyme substrate complex. We have our substrate, we have our enzyme. It changes shape. And then at the end, we have our products. Then this enzyme can go ahead and look for another substrate to break apart. Here's an example of an enzyme synthesis reaction. So like we said, not all enzymes break apart molecules. Some enzymes are used to make molecules. So as we can see in this enzyme needs substrate 1 and substrate 2. Substrate 1 and substrate 2 bind together at the active site. In this instance, the substrate binds to form the product, and at the end, we have a product that's being made, not one that's being broken apart. Here are a few enzyme videos to help you guys out. So we have the Amoeba Sisters. We have just a very basic enzyme introduction, how digestive enzymes work, and then we have our Bozeman Bio video as well, which gets into a little bit more in depth. Please watch these videos to help you guys understand the material a little bit more clear. All right. What are some properties of enzymes? What are some of the properties of the enzymes? So the specificity of the enzymes. These are reaction specific, meaning that each enzyme is substrate specific due to the fit between the active site and the substrate. The substrates are held in the active site by weak interactions, held together by hydrogen bonds and ionic bonds. Enzymes are named for the reaction they catalyze, and as you notice, most enzymes end in ASE. So if it ends in ASE, ASE equals an enzyme. Okay. If it ends in OSE, OSE is going to be a sugar. Okay, OSE is a sugar. So let's look at some of these enzymes. Sucrase breaks down sucrose. Proteases break down proteins. Lipases break down lipids. DNA polymerase builds DNA. And pepsin breaks down proteins. As we said, enzymes are reusable. They're not consumed in the reaction. So a single enzyme molecule can catalyze thousands of more reactions per second. The enzymes are unaffected by the reaction, as we see here. So enzyme's going to attach to the substrate. It's going to break it apart to form the products. And once it's done, it's going to go ahead and look for another substrate and do the same reaction over and over and over again. Now, there are some factors that affect enzymes. These are factors that affect enzymes. Please make sure that you know these. So enzyme concentration, meaning the amount of enzymes. Substrate concentration, meaning the amount of substrates. Temperature, pH, salinity or the salt content, activators, and inhibitors. So let's look at these individually. All right, enzyme concentration. What does this mean here? There's going to be a lot of graphs that you guys are going to have to understand for this unit. It's a lot of graphs. All right, so what this states here is if I increase the enzymes, I increase the reaction rate. So the more enzymes I have, the more substrates are going to be broken down. So the effect on rates of enzyme activity. As I increase the enzymes, I increase the reaction rate, meaning more enzymes equals more frequently collide with the substrate. The reaction rate will level off, meaning that the substrate becomes the limiting factor and not all enzyme molecules can find the substrate. So going back, eventually once there's no more substrate to break apart, this reaction here then starts to level off. All right, substrate concentration. So let's say I infiltrate your body with a whole lot of substrate. Well, if there's more substrates, my reaction rate is going to increase, same thing, until it starts to level off. So the effect on rates of enzyme activity. As I increase the substrate, I increase the reaction rate. More substrates equals more frequently collision with the enzymes. The reaction rate levels off, and then all enzymes have active sites that are engaged. The enzymes are saturated because there is a maximum rate of reaction, meaning that there's more substrate. So the more substrate it is, the more likely an enzyme is going to bounce into it and break it apart. So that means that the reaction rate is going to increase. It's going to increase until once again there are no more substrates. Temperature. Now notice here with temperature. When I increase the temperature, I increase the rate of the reaction until I hit this top part, which is called the optimum rate. Once I hit the optimum rate, now notice that my reaction rate starts to decrease. 37 degrees Celsius is our body's optimum rate at which it functions. So the effect on rates of enzyme activity. Optimum temperature. The greatest number of molecular collisions occur between 35 to 40 degrees Celsius or 98.6 degrees Fahrenheit. Now, if I increase beyond the optimum temperature, what happens is the increased agitation of molecules disrupts the bonds. The bonds are broken, and now the enzyme starts to unfold or it denatures. So it loses a 3D shape. Basically, the tertiary structure in our enzymes or in our proteins start to break apart. So the enzyme disrupts the hydrogen bonds, which forms the protein itself. As it starts to disrupt the bond, it denatures and it loses shape. However, if I decrease the temperature, so let's say, for example, I put these enzymes in cold temperatures, the molecules move slower and it decreases the collision. That is why we put food in the refrigerator. Food goes into the refrigerator to stop it from spoiling. So what's being spoiled? Basically, as I leave food on the counter, different enzymes or different bacteria fall on the food, right? Over time, those bacteria then start to eat on the food that it fell on. And then it starts to denature, then it starts to break down the food. So in order for us to stop that from occurring, we put the food inside of a refrigerator where the bacteria cannot grow as efficiently. And so in that instance, it stops the food from spoiling. Now, different enzymes function in different organisms. So, for example, if I look at these two different enzymes, so depending on who you are, like we said, this enzyme here is going to be working best within this temperature. This one here, the optimum temperature for the enzymes for hot springs prokaryotic organisms is at 70 degrees Celsius. So, the enzymes are going to work better depending on what their environment is. So as we can see here, pH is another factor that affects enzymes. There's three different pHs that we are going to look at that work in our systems. It's going to be a lower pH, a pH at 7, and a pH above 7. So one that works as an acid, one that works in a base, and one that works in a neutral environment. All right, pH, the effect on rates of enzyme activity. So pH changes the protein shape. The attraction of the charged amino acids is going to cause the enzymes to denature. The pH changes charges. So when I add or I move a hydrogen, it disrupts the bond once again and it causes the protein to denature. As we said, most human enzymes work between a pH of 6 to 8, and it depends on the localized conditions. Pepsin works in a pH of 3, whereas trypsin works in a pH of 8. So salinity. Salinity is the salt concentration located within our systems. So salt also affects the way an enzyme functions. Notice here's our optimum rate. Same thing as I increase the salt concentration, I increase the rate of the reaction until I hit the optimum rate. And then after that, the enzyme starts to unfold and it denatures. So the effective rates on enzyme activity. How does salt affect the protein shape? It depends on the attraction of the charged amino acid side. The salt changes the charges. So we either have more positive or more negative ions. These then once again disrupt the bonds and it disrupts the 3D structure, basically the tertiary structure of the protein. So once again, if I disrupt the structure, it no longer functions. So the enzymes are intolerant of extreme salinity. So that's why the Dead Sea is called dead for a reason. It's high in salt. So because it's so salty in nature, nothing can live inside of it. Enzymes don't function within the high amounts of salts. Some enzymes also need what are known as activators. And what an activator does is it's a compound that actually helps the enzyme. It helps the enzyme to function. Hence the term activate. There's two different types of activators that we're going to talk about. The first ones are known as cofactors, and the second ones are known as coenzymes. So cofactors and coenzymes. So cofactors are non-protein and they are small inorganic compounds, inorganic. So if they're inorganic, that means that they don't contain carbon. An example of some cofactors are the elements magnesium, potassium, calcium, zinc, iron, and copper. And what these inorganic molecules do is they bind to the enzyme and they activate it so that it functions. A good example is iron, which is found in hemoglobin. Hemoglobin is the protein which is found in our blood. And what it does is it helps to carry oxygen. Now, iron is necessary to activate the hemoglobin protein. If iron levels are low, then that means that it doesn't carry the oxygen very well in our systems and we can become anemic. Okay. Another example, like we said, are coenzymes. Coenzymes are non-protein, but these are organic molecules, meaning that they contain carbon, and they bind temporarily or permanently to the enzyme near the active site. So many vitamins such as NAD or niacin, FAD, which we find in riboflavin, as well as our coenzyme A. Sometimes enzymes have what are known as inhibitors. And what inhibitors do is exactly what the term means. It inhibits. So an inhibitor is going to stop an enzyme functioning. It's going to stop an enzyme from functioning. So regulation of enzyme activity and other molecules that affect the enzyme activity. Now there's different types of inhibitors we have. We have selective inhibition and activation. So the 4 types we are going to look at is competitive inhibition, non-competitive inhibition, irreversible inhibition, and feedback inhibition. So once again, an inhibitor stops or it limits the enzyme from working. All right, what are competitive inhibitors? Competitive inhibitors. So the effect. An inhibitor and a substrate compete for what is known as the active site, which is known as the active site. So an example would be penicillin. Penicillin blocks the enzyme that the bacteria use to build the cell walls. So as I can see here, if I take penicillin, then what it does is it's going to attach here to the active site. If it attaches to the active site, then it does not allow the substrate to bind. Why? Because I changed the shape of the enzyme. If I change the shape, it's not going to function. Okay. So how do we overcome these? So we overcome competitive inhibitors by increasing the substrate concentration. So you saturate the solution with the substrate so that it outcompetes an inhibitor for an active site on an enzyme, Okay. All right, non-competitive inhibitors. Anything that is a non-competitive inhibitor means that it's an inhibitor, but it binds to any other site other than the active site. So notice here in this non-competitive inhibitor, also known as an allosteric inhibitor. Allosteric is the same as non-competitive. They're synonymous, okay? And what this does is this inhibitor attaches to a site other than the active site. So notice how it attached here. Now when it attaches here, it does a conformational change of the enzyme, which now the enzyme will not be able to allow the substrate to attach because it's changed the shape. So it's not able to bind. So these are known, like we said, as allosteric inhibitors. Some examples are some anti-cancer drugs inhibit the enzymes involved in synthesis and nucleotides and therefore in building DNA. So if I interfere with the development of DNA, then I stop DNA production and I stop division of more cancer cells. So these types of anti-cancer drugs, once again, they attach to the enzyme where DNA is being made, and then they inhibit DNA production, or they inhibit the cells from reproducing. Heavy metal poisonings are the same example, as well as cyanide poisoning. Cyanide poisoning attaches to the enzymes and can cause death. So these causes the enzymes to change shape, and they renders the active site unreceptive. Once again, it changes the shape of the active site. All right, in irreversible inhibition, this is when the inhibitor is permanently bind to the enzyme. It permanently binds. So if it's a competitor, it's going to permanently bind to the active site. And if it's an allosteric, it's permanently going to change the shape of the enzyme. So some examples are like nerve gas, sarin, and many other insecticides. These here can permanently change the shape of the enzyme. So cholinesterase is inhibitors. They don't break down the neurotransmitter acetylcholine. So these are what we find in like snake venoms All right, so as we can see here are our inhibitors and our activators. So our regulatory molecules attach to the allosteric site, causing conformational change in the shape. The inhibitor keeps the enzyme in an inactive form. And as we could see, the activator keeps the enzyme in the active form. Right, enzymes also play an important role in metabolic pathways. Sorry about that. In metabolic pathways. And what are metabolic pathways? Basically, these are chemical reactions of life. They're organized in these certain pathways. So what they do is they divide the chemical reaction into many smaller steps. And this is to make sure that the steps are more efficient and to control what's being made. So for example, I have my substrate A. At the end, I need to make substrate G. So I'm going to have 1, 2, 3, 4, 5, 6 different enzymes to get here. So A is going to turn to B is going to turn to C is going to turn to D is going to turn to E is going to turn to F, and then F turns to G. So what would happen if my enzyme 5 was not functioning? Enzyme 5 is not functioning, E cannot change to F. If F can't change to G, then I can't make the product. So this is a metabolic pathway. This is a metabolic pathway. Now, these groups of enzymes are organized. So if the enzymes are embedded in a membrane, they are arranged sequentially. They link endergonic and exergonic reactions, just like the production of ATP and ADP. That is an endergonic and exergonic reaction. In this example here, we're looking at the cell membrane and how these series of proteins help to make our ATP, as we said. All right, feedback inhibition. This is the regulation and coordination of the production. Like we said, the product is used by the next step in the pathway. So the final product is inhibitor of the earlier step. Our allosteric inhibitor of the earlier enzyme will stop our feedback inhibition. So no unnecessary accumulation of the product is being released. Okay. So for example, once I'm done making G, then what will happen is there's going to be a button or a signal that's going to stop enzyme 1 from being made. If I stop enzyme 1 from being made, then notice that I can't go through this series of chemical reactions to get G, okay? All right, and then the last example here, as we see, is the synthesis of our amino acid isoleucine from the amino acid threonine. So once again, in order to get this end product, I need these series of different types of enzymes to translate this particular protein. Well, that'll do it for all of our enzymes. I'm hoping by the end of this section, you should know the structure and function of enzymes. You should know what are some properties that inhibit or that alter enzymes from functioning. And you should be able to understand the different types of inhibition models. Hoping you guys have a great day. Bye.